Welcome to the Deep Dive, where we cut through the noise to bring you the essential insights on complex topics. When you hear gastric cancer, your mind might immediately go to the most common type, tubular adenocarcinoma, which, yes, accounts for a large majority, maybe up to 72% in places like South Korea. But what if you knew that beneath this common understanding lies a whole hidden universe of other gastric cancers? One so rare, so incredibly unique, they present entirely different puzzles for doctors, researchers, and ultimately patients. Today, our deep dive is into this often perplexing world of unusual or uncommon histologies of gastric cancer. We're going to unpack a recent review that spotlights these lesser-known variants, shedding light on their distinct characteristics, the diagnostic hurdles they pose, and crucially, why understanding them is so vital for providing the right patient care.
It's fascinating, really, how these rare forms well, they defy typical presentations. They force us to rethink what we thought we knew about stomach cancer, often creating these diagnostic conundrums and therapeutic complexities precisely because their unique pathological features are so difficult to categorize. Our mission today is to extract the most important nuggets of knowledge from this material, helping you grasp these intricate distinctions from their surprising behaviors to what they actually look like at a microscopic level.
So what exactly does this mean for the patient? We're talking about a spectrum of diseases with very distinct features under the microscope.
Right.
Yet they often have overlapping clinical presentations, making clear-cut diagnoses incredibly challenging.
Exactly. And because cases are so limited, their unique nature and how they develop aren't fully understood.
But despite their rarity, recognizing these types can quite literally make all the difference in a patient's journey. Let's jump into some of these specific, unusual variants. Okay, let's kick things off with papillary adenocarcinoma. The name gives us a clue, but what makes this particular variant so distinct and often so challenging for patients?
Well, imagine these tumors under a surgeon's eye. They often appear as distinct, sort of mushroom-shaped growths protruding from the stomach lining. But the real story is under the microscope. Picture tiny, well-organized, finger-like projections, almost like a miniature coral reef. You can see this clearly in Figure 1. These fingers are lined by neat column-shaped cells that maintain their structure, all supported by delicate connective tissue cores, these fibrovascular cores. Sometimes you might even see tubular structures interwoven, giving it a combined tubulo-papillary appearance. This distinct finger-like structure, as shown in the figure, is what defines them.
And what about the outlook for patients with this type? Does that unique structure have implications?
It absolutely does. The key insight here isn't just its rarity making up about, say, 2.7% to maybe 9.9% of gastric cancers, but its unusual predilection for liver metastasis.
Ah, the liver specifically.
Yes, that specific pattern of spread is a critical red flag for clinicians. It guides specialized imaging and treatment strategies that differ significantly from other gastric cancers. Unfortunately, this tendency to spread to the liver directly leads to poorer survival rates. It's also worth noting that some rare forms, like clear cell variants, which have this glycogen-rich clear cytoplasm, can even produce alpha-fetoprotein, or AFP.
AFP, isn't that usually linked to liver cancer or fetal development?
Precisely. So when AFP shows up unexpectedly in stomach cancer, like in some papillary types, or what's called gastric adenocarcinoma with interblastic differentiation, GAED, it can signal a particularly aggressive form, although GAED itself might have a slightly better outlook than other AFP producers.
That's a sobering outlook for papillary adenocarcinoma. Now, if that was concerning, our next variant, micropapillary carcinoma, introduces an even more aggressive challenge due to a peculiar microscopic feature. Tell us about it.
Indeed. This type carries an even worse prognosis, primarily because it's associated with much higher rates of spread to the lymphatic system and lymph nodes, lymphovascular invasion. If you were to peer through a microscope, as shown in Figure 2, you'd see something quite unusual. Tiny clusters or nests of tumor cells. But unlike most cancers, they surprisingly lack the usual connective tissue scaffolding or fibrovascular cores.
No support structure.
Exactly. Instead, these small nests seem to float freely within transparent, empty-looking spaces, these lacunar spaces. It almost looks as if they're mimicking lymphatic or vascular channels themselves. This free-floating appearance, lacking those cores, is precisely why they're thought to be so aggressive and prone to spreading quickly through the lymphatic system. And this micropapillary component, it can actually be found within other conventional tumors, like tubular or papillary adenocarcinoma. Its proportion can vary significantly, from just 5% to maybe 90% of the whole tumor.
So it's those tiny, unsupported clusters and are the real troublemakers. Let's shift gears now to a cancer that's a true blend, adenosquamous carcinoma. This sounds like it combines two distinct types of cancer in one.
Precisely. This is a primary carcinoma uniquely characterized by exhibiting both glandular components, like typical adenocarcinoma, and squamous cell elements. Think skin-like cells. To be classified as adenosquamous, the squamous part must make up at least 25% of the tumor volume.
Okay, a significant chunk then.
Yes. Clinically, these tumors often present as large masses, most frequently found in the lower third of the stomach. Histopathologically, the squamous component is quite distinct. You can see it in Figure 3A. It features something called keratin pearl formation, tiny concentric rings of tough keratin protein and intercellular bridges, which are visible connections between the cells. And this is all alongside the glandular adenocarcinoma elements. Pathologists use special stains, like P63 or P40, to confirm the presence of these squamous cells. Figure 3B shows this, where those squamous components light up positive for P40.
So what does this aggressive combination mean for the patient?
Well, these tumors carry a poor prognosis. This is due to their tendency for deep penetration into the stomach wall and a high propensity for invading blood and lymphatic vessels. This often means they're diagnosed at a more advanced stage, with a higher likelihood of widespread metastasis. The dual nature also makes them notoriously difficult to treat, as therapies effective for one component might not work as well for the other.
And even rarer than adenosquamous carcinoma is pure gastric squamous cell carcinoma, or SCC. You mentioned fewer than 100 cases ever reported.
That's astonishingly rare, yes.
Yeah.
Accounting for a tiny fraction, maybe 0.04% to 0.07% of all gastric cancers. What defines it microscopically, as you can see in Figure 4, are these keratinocyte-type cells. Imagine them as tough, skin-like cells, exhibiting features like those same intercellular bridges and keratinization we just discussed.
Okay.
But crucially, lacking any of the glandular components seen in adenosquamous carcinomas. It's like finding a patch of skin cancer, essentially, growing in your stomach, completely independent of the typical stomach lining cells.
Where does it even come from?
The exact origin isn't definitively known, but hypotheses include arising from squamous metaplasia, where cells change their type within existing adenocarcinomas, or maybe from very early multipotent stem cells. Prognosis is generally poor, as it's often diagnosed at an advanced stage, perhaps because its unusual nature makes it harder to suspect early on.
This next one sounds truly fascinating. Hepatoid adenocarcinoma, or HAC. The name itself suggests a crossover with the liver. How does a stomach cancer get a name like that?
It absolutely does mimic liver cancer. This gastric cancer is characterized by tumor cells that strikingly resemble hepatocellular carcinoma, or HCC, the most common type of liver cancer. Under the microscope, as shown in Figure 5A, you'd see these large polygonal tumor cells with a distinctive pink granular cytoplasm, eosinophilic cytoplasm. They're often arranged in patterns like trabecular or acinar structures, almost identical to those found in the liver. A key identifier is alpha-fetoprotein, AFP production, which we mentioned earlier.
Right, the AFP again.
Yes, it's often found elevated in both blood and the tumor tissues of patients with HAC, which adds to the liver-like confusion. Figure 5B actually shows the tumor cells testing positive for AFP using immunohistochemistry.
That's incredible. A stomach cancer that looks and acts like a liver cancer. How do pathologists definitively tell them apart, and what's the takeaway on its aggressiveness?
It's a real diagnostic challenge, like a master mimic. While they look alike, pathologists use special immunohistochemical stains. HAC typically shows only focal staining for a marker called HepPAR1, whereas true HCC usually has extensive staining. Plus, HAC often has significantly higher rates of cytokeratin 19 and CK20, which are markers more common in gastric cancers.
Okay, so there are clues.
There are. As for aggressiveness, this disease is definitely aggressive. It frequently infiltrates blood vessels and leads to high rates of metastasis, particularly to the liver and lymph nodes. This unfortunately results in a poorer prognosis. It's worth noting, though, that not all AFP-expressing gastric carcinomas are HAC. We mentioned GAED before, gastric adenocarcinoma with enteroblastic differentiation. That also produces AFP, but tends to have a relatively more favorable prognosis compared to HAC. GAED typically has a tubular papillary structure, clear cytoplasm, reminiscent of fetal gut tissue, and expresses different markers like CEL-O4 and glypican-3, which help differentiate it.
Speaking of rare and unique, gastric choriocarcinoma sounds like something from another part of the body entirely, placental tissue.
It is quite startling. This variant exhibits both adenocarcinomatous differentiation, meaning it has gland-forming cancer cells, and germ cell elements, specifically cells called syncytiotrophoblasts and cytotrophoblasts, which are, yes, typically found in the placenta during pregnancy.
Wow.
Imagine seeing placental cells growing in a stomach tumor. Figure 6a illustrates this combination. The pathogenesis is thought to involve a complete choriocarcinomatous transformation of a conventional adenocarcinoma. Macroscopically, when a surgeon sees it, it often presents in a bulky exophytic mass with significant areas of dead tissue, necrosis, and hemorrhage. A critical marker is human chorionic gonadotropin, or HCG, the hormone detected in pregnancy tests.
HTG, seriously?
Yes, its presence in the trophoblastic components, often markedly elevated in the patient's serum, is a key diagnostic clue. Figure 6b shows these tumor cells staining positive for beta-HCG.
And the prognosis for such an unusual and aggressive type?
Management is incredibly challenging due to its highly aggressive behavior and complex histology. It frequently metastasizes through the bloodstream hematogenously, often leading to liver failure with rapid and widespread dissemination. This significantly impacts prognosis. Sadly, patients often survive only a few months post-diagnosis. It truly is one of the most aggressive forms we encounter.
Moving to something with a very different character, gastric carcinoma with lymphoid stroma, or GCLS, also known as lymphoepithelioma-like carcinoma. That sounds like it has a strong immune component.
You're spot on. This uncommon subtype, accounting for maybe 1% to 8% of gastric cancers, is histologically quite diverse. It can range from well-defined cellular patterns like trabeculae to less organized syncytial structures. But its defining feature, and what makes it so distinctive, is a dense infiltration of immune cells, specifically lymphoid cells, primarily cytotoxic CD8-positive T cells, woven right into the tumor architecture. Figure 7a gives you a sense of this density.
So the immune system is right there in the fight.
It appears so. It's like the body's immune system is actively present within the tumor itself. A significant aspect of GCLS is its strong association with the Epstein-Barr virus, EBV, the virus that causes mono. Positivity rates for EBV can be as high as 80% or more, often detected using a test called EBER, in situ hybridization, which you can see in Figure 7b highlighting the tumor cells, not the lymphocytes. EBV is thought to contribute through specific molecular events, like altering DNA methylation patterns. Interestingly, there's also an EBV-negative subset of GCLS cases that have been identified as having high microsatellite instability, or MSIH, which is linked to defects in DNA mismatch repair.
This raises an important question. Does this strong immune presence impact the patient's outlook? Does it mean the body is putting up a better fight?
Yes, it seems to. GCLS predominantly occurs in the upper part of the stomach, the proximal stomach, and is more common in males. Importantly, it's often diagnosed at an earlier stage than other gastric cancers. This early detection, combined with that robust associated immune response, likely contributes to a comparatively improved prognosis.
That's actually good news for a change.
It is. And what's even more fascinating, potentially, is that these tumors frequently overexpress immunomodulatory molecules like PD-L1 and PD-L2. This suggests they could be promising targets for cutting-edge immune checkpoint inhibitors, therapies designed to boost the immune system's attack on cancer.
So harnessing the body's own defense system.
Exactly. This makes GCLS a potentially really interesting area for treatment development.
Okay, next up. Carcinosarcoma of the stomach, another one that sounds like a truly mixed bag of different cancer types. Sarcoma components.
Precisely. This is a biphasic malignancy, meaning it features both adenocarcinoma, epithelial cancer, and sarcoma, connective tissue cancer, components within the same tumor. The sarcomatous part often displays a spindle cell morphology. The cells look long and thin like spindles, and it can strangely resemble various types of sarcomas, like osteosarcoma bone, chondrosarcoma cartilage, rhabdomyosarcoma muscle, or even leiomyosarcoma smooth muscle, right there mixed in the stomach tumor. Some cases even show neuroendocrine or adenosquamous differentiation, highlighting their remarkable heterogeneity. It's a real mix.
That's a truly aggressive blend. How does it typically present, and what are the challenges?
Most gastric carcinosarcomas appear as large, polypoid or fungating masses, bulky tumors. Their aggressive behavior and the advanced stage at which they are often diagnosed contribute to a generally poor prognosis. A key challenge for pathologists is distinguishing true carcinosarcomas from benign bone formation, ossification, or osteoid production that can occasionally occur in gastric carcinomas as a reaction. It requires careful evaluation to ensure it's actually a cancerous sarcomatous component, not just, some benign bony bits in the stroma.
Gastroblastoma is a unique neoplasm, particularly because it primarily affects children and young adults, which is unusual for stomach cancer.
That's a key distinguishing feature, yes, along with a slight male predominance. This is another biphasic tumor composed of mixed spindle and epithelial cellular elements. But importantly, these elements lack sufficient abnormality or atypia to be diagnosed as the more aggressive carcinosarcoma we just discussed. They look a bit more uniform. Under the microscope, as shown in Figure 8, the epithelial cells have scant pale cytoplasm and round nuclei, while the spindle cells are monotonous and slender, often sitting in a sort of myxoid or jelly-like background. These tumors are most commonly found in the antrum, the lower part of the stomach.
Are there any unique molecular markers for gastroblastoma that help in diagnosis or perhaps treatment?
Yes, a defining molecular feature has been identified: the presence of the MEL-AT1-GLI1 fusion gene.
A fusion gene.
Yes, think of it as two separate genes abnormally joined together, creating a specific genetic switch that drives the cancer's growth. Both the mesenchymal, spindle cell, and epithelial components of the tumor are strongly and diffusely positive for GLI1 immunolabeling, which is a powerful diagnostic tool.
And the outlook.
Prognosis for gastroblastomas is generally favorable following surgical resection, which is good news, especially for young patients. Though regional and distant metastases, while rare, have unfortunately been reported.
Let's talk about parietal cell carcinoma and oncocytic adenocarcinoma. These sound like they're related to specific cell types in the stomach lining itself, the ones making acid.
They are indeed. Parietal cell carcinoma strikingly resembles the acid-secreting parietal cells normally found in the gastric lining. It has that characteristic eosinophilic granular cytoplasm, very pink and grainy looking. Histologically, it grows in solid sheets of cells, though some might form small gland-like clefts. Special stains can highlight the cytoplasm, and crucially, the cells test positive for parietal cell-specific antibodies, like H plus K plus ATPase, which is the proton pump, the acid pump.
Okay, so it really looks like the acid cells. What about oncocytic?
Oncocytic adenocarcinomas share many features: abundant pink, granular cytoplasm, packed with numerous mitochondria, the powerhouses of the cell. That's what makes them look granular and pink, oncocytic. However, a crucial distinction is that they do not express the parietal cell-specific H plus K plus ATPase.
Ah,
So while they look similar due to all the mitochondria, they lack that specific parietal cell marker. Some experts refer to these as mitochondria-enriched carcinomas.
Given their extreme rarity, what's known about their prognosis, and is their classification clear-cut?
Well, they are exceedingly rare. Parietal cell carcinoma is generally assumed to have a more favorable prognosis than typical gastric carcinomas, and mitochondria-enriched carcinomas also tend to have a favorable outlook, often being small and detected at an early stage with infrequent lymph node metastasis. The challenge here lies in their classification and terminology. There's still quite a bit of overlap and, frankly, ambiguity among parietal cell, oncocytic, and mitochondria-enriched carcinomas. We really need more cases and research to establish clear boundaries. It's also important to note that gastric adenocarcinomas of the fundus gland type, which we'll get to, while also involving parietal cells, are distinct from these. They are typically small, well-differentiated, and lack that solid, sheet-like growth pattern.
Okay, moving on to Paneth cell carcinoma, another rare one, tied to a specific cell type known for its defensive role in the gut.
Indeed. This is characterized by a predominance of Paneth cells. These are cells typically found at the base of crypts in the small intestine and parts of the colon. They're known for secreting antimicrobial peptides, basically, acting as part of the gut's innate immune defense.
So finding them dominating a stomach cancer is unusual.
Very. These cancer cells have very distinctive eosinophilic or pink staining cytoplasmic granules and test positive for markers like lysozyme and defensin 5, which are characteristic of Paneth cells. It's hypothesized that Paneth cell carcinoma might develop from pre-existing lesions in the stomach that have undergone intestinal metaplasia, meaning stomach cells started to look and act like intestinal cells, including having Paneth cells. Because it's so extremely rare, our understanding of its clinical presentation and effect on patient survival is very limited. Occasionally, you might just see some Paneth cells scattered within a typical gastric adenocarcinoma, but that's different from this distinct carcinoma type.
Our next variant, gastric adenocarcinoma of fundus gland type, or GAFG, seems to be another well-differentiated type, but how does it distinguish itself? Sounds like it mimics the stomach's own glands.
Yes, exactly. It's a well-differentiated neoplasm that actually develops from a benign growth called an oxyntic gland adenoma. It accounts for a small percentage, around 1% of early gastric adenocarcinomas. Its hallmark is differentiation that closely resembles the oxyntic glands, which are the main acid-producing glands of the stomach body and fundus. These are classified into chief cell predominant type, which is the majority, parietal cell predominant type, and mixed types. Under the microscope, as you can see in Figure 9, you'd see well-differentiated column-shaped cells with pale, sort of basophilic cytoplasm and only mild nuclear abnormalities or atypia. They look quite organized.
It's relatively slow growing.
Generally, yes. While submucosal invasion growing just beneath the surface layer is seen in a significant proportion of cases, maybe 60%, most lesions are detected at early stages. And importantly, lymph node metastasis is fortunately rare. They are positive for specific markers like pepsinogen I, made by chief cells, and MUC6, and those with parietal cell differentiation will, again, be positive for H plus K plus ATPase, the acid pump. So it's typically a relatively slow-growing, early-stage cancer that closely mimics the normal gland structure.
This next one sounds like one of the most challenging types to diagnose. Undifferentiated carcinoma. What makes it so difficult to pin down?
It absolutely is one of the toughest. These are characterized by highly abnormal anaplastic cells that shockingly lack any specific cytological or architectural differentiation.
Meaning they don't look like anything recognizable.
Exactly. They don't resemble glands or squamous cells or really any specific cell type or structure. Figure 10a shows this lack of differentiation. This makes them notoriously heterogeneous and incredibly difficult to distinguish from other aggressive malignancies like lymphomas or sarcomas, even for experienced pathologists. Microscopically, the tumor cells appear chaotic, disorganized, maybe sheet-like, but without clear features. To confirm their epithelial nature, immunohistochemistry is critical. They usually show positive staining for cytokeratin, a marker of epithelial cells, which helps rule out lymphoma or sarcoma. Interestingly, they also often show co-expression of vimentin, which is typically a mesenchymal marker, adding to the confusion.
So if they're so undifferentiated, what causes them to lose all their normal characteristics? Is it known?
Well, some cases suggest they might originate from other more differentiated gastric cancer types through a process called dedifferentiation.
Losing their identity, basically.
Yes, where cells lose their specialized features and revert to a more primitive aggressive state. A significant finding in recent years that underlying genetic alterations can involve components of the Swiss-MF chromatin remodeling complex. Think of this complex as the cell's internal architectural firm that helps organize its DNA and control gene expression. When key proteins in this complex, like SMARCB1, also called INI1, SMARCA4 or BRG1 or ARID1A, are lost due to mutations, the cell loses its proper regulation, leading to uncontrolled, highly undifferentiated growth. For instance, Figure 10b shows an example of a SMARCA4 mutant carcinoma, where the BRG1 protein staining is lost in the tumor cells, indicating that deficiency.
And the prognosis.
Given their extreme rarity and highly aggressive nature, the prognosis is generally poor. They progress rapidly, often with high mortality within just a year of diagnosis, and frequently present at advanced stages. It's one of the most unforgiving variants.
And finally, extremely well-differentiated adenocarcinoma, or EWDA. The well-differentiated part sounds encouraging, almost benign, but I'm guessing there's a diagnostic catch.
There is a huge catch, and it's a major diagnostic challenge. EWDA is characterized by nuclear features that appear quite non-aggressive, almost normal-looking. The key is subtle architectural abnormalities, things like glands that are unusually twisted, tortuous, branching in odd ways, or slightly enlarged, distended. It comes in intestinal and gastric types, resembling either intestinal metaplasia or hyperplastic foveolar epithelium, respectively both relatively common, benign-looking changes.
So it looks like something harmless.
Exactly. The major challenge is its innocuous appearance. It looks so benign that it's often misinterpreted as just regenerating tissue after injury or inflammatory changes, especially in small endoscopic biopsies. The true hallmark, the critical clue that pathologists need to look for, is those structural abnormalities, the unusual branching or distortion of the glands, not significant cellular atypia, which is what usually flags cancer. Endoscopically, these can appear as ill-defined, flat, or slightly depressed lesions without significant color changes, making them easily missed by the endoscopist too.
Wow, hiding in plain sight. What about prognosis?
Well, generally having a favorable prognosis, because it is, after all, extremely well differentiated, there have been reports of some cases evolving into a more aggressive diffuse type. So careful diagnosis and long-term follow-up are still crucial. It really highlights the need for pathologists to be aware of these subtle architectural clues. So if we try to connect all this to the bigger picture, it becomes abundantly clear that gastric cancer is definitely not a singular disease. It's a complex landscape with numerous histological subtypes. Our deep dive today has highlighted just how incredibly diverse these forms can be, from the core-like papillary to the floating micropapillary, the dual nature of adenoid squamous, the liver-mimicking hepatoid, and even the immune-rich GCLS.
Yeah, it's quite a spectrum, and each of these subtypes presents unique diagnostic challenges and distinct pathological features that demand incredibly careful evaluation. What really stands out, and I think what's crucial for you listening to take away, is that understanding these rare variants isn't just an academic exercise. It's absolutely fundamental for accurate diagnosis, for tailoring effective treatment planning.
Right.
And ultimately, for dramatically improving patient outcomes, getting the diagnosis right for these rare types is critical.
Absolutely. And this understanding really underscores why ongoing research and continuous case studies are so important. They deepen our knowledge of these uncommon cancers, paving the way for potentially more effective treatments and certainly better prognostic assessments. Recognizing the diversity of these histological characteristics is key because these unique features directly impact every single decision made in patient care and directly influence prognosis.
That was a truly insightful deep dive into the hidden complexities of gastric cancer. It's a powerful reminder that even within diseases we think we know well, there's always more to learn and uncover.
Indeed. And it raises an important question as we wrap up. What might further research into these rare gastric cancer variants reveal about the fundamental mechanisms of cancer development itself, not just in the stomach, but maybe more broadly? Could these specific insights, gleaned from these uncommon forms, potentially unlock completely new therapeutic approaches for other challenging malignancies down the line?
Exactly. That's a fascinating thought to mull over as you continue your own deep dives into the world around you. Thank you so much for joining us for this profound exploration.